Assalamu alaikum friends. Today we have here to discuss the TCA cycle, tricarboxylic acid, also known as Krebs or citric acid cycle. What's the function of this citric acid cycle is basically convert the acetyl oxidation of this acetyl-CoA to carbon dioxide. It's occur in the this whole process occur in the mitochondria. Now we have discussed step by step this TCA cycle. Now here we can see that there is the pyruvate which is formed by a glycolysis which we discussed earlier. This pyruvate is converted into an acetyl-CoA which is basically a 2 carbon unit. It is done by a pyruvate dehydrogenase enzyme which is a rate limiting enzyme, and it's controlled by many coenzymes. Now these 2 unit carbon acetyl-CoA with oxaloacetate which is a 4 carbon unit combined to form a citrate and done by a citrate synthase enzyme. Now what's the importance of this citrate is basically it's a intermediate for the fatty acid synthesis by means of citrate shuttles. Now this citrate is converted into isocitrate by a cis-aconitase enzyme. This enzyme is not much important to remember that. Now this isocitrate is converted into an alpha ketoglutarate. Now it's a very important enzyme and we should know for a board exam. Now isocitrate dehydrogenase, as name suggests that here the NADH is converted into an NADH, and plus the 1 carbon is also removed and decarboxylation reaction also occur here. So from 6 carbon unit is converted into a 5 carbon unit. This enzyme is activated by ADP and plus inhibited by NADH and ATP. Now this alpha glutarate is converted into succinyl-CoA by another important enzyme known as alpha glutarate dehydrogenase. So the name suggests that dehydrogenase reaction occur in which the NAD is converted into NADH and another decarboxylation reaction occur in which the one other carbon atom is removed. So now this 5 carbon unit is converted into a 4 carbon unit. What's the importance of this enzyme is that it is a multiple enzyme complex. For mnemonics we have right here that tender loving care for Nancy. So tender, so T means it's required thymine, L for lipoic acid, C for coenzyme A, F for FADH and N for NADH. And another important for the succinyl-CoA is that it's the intermediate for heme synthesis plus activates ketone bodies in extrahepatic tissue. Now this succinyl-CoA is converted into a succinate by a succinyl-CoA synthase enzyme. What's the important here? From here the GTP is converted into a GTP. Now from here the succinate is converted into a fumarate by a succinate dehydrogenase enzyme, which is also known as complex 2 because it helps in electrons transport chain, which will be discussed earlier. Now here the FAD is converted into FADH. Now the fumarate is formed. The fumarate is converted into a malate by a fumarase enzyme. What's the importance of malate is that it is a part of gluconeogenesis, which will be discussed in the chapter of gluconeogenesis. Now from malate, which is a 4 carbon unit, is converted into oxaloacetate by a malate dehydrogenase enzyme in which the NAD is converted into NADH and the whole cycle is gone. Now what's important from here that how many ATPs are formed from this citric acid cycle. So we know that the NAD and NADH when the NADH is converted to NADH, so here we can form the 3 ATPs. So we have right here that 3 ATP. Now we also 3 from here from isocitrate dehydrogenase and 3 from alpha ketoglutarate dehydrogenase. Now when the GTP is converted into GTP, so here we can gain only 1 ATP. When the FAD is converted to FADH, so we form 2 ATPs. So here again 3 ATP. So total we have 3, 6, 7, 8, 9 and the 12. So 12 ATPs form from citric acid cycle. What's the other importance are that the succinate dehydrogenase is the only enzyme which occur in inner mitochondrial membrane, and the all other occur in the matrix of the mitochondria. So for more videos, please like my page and subscribe my page. Thank you.